So, once again, very good afternoon. So, today I will deliver a talk on target of rapamycin signaling, key signaling pathway for starch accumulation in red alga. So, before starting a talk, I will briefly explain about the organization from
Yes,
where I am.
Gopal.
So, actually, I am assistant professor in the Department of Industrial Biotechnology at Gujarat Biotechnology University. As Poonam Madam mentioned, this is the initiative of Government of Gujarat DST India. So, we are offering a Master of Biotechnology in a 5 different disciplines. That is the industrial biotechnology, environmental biotechnology, medical biotechnology, animal biotechnology, and plant biotechnology. We have also recently initiated the PhD program, and our university is located in the beautiful gift city, Gandhinagar. So with this, I will start my talk. So as we all know that because of the various issue, our Mother Earth is experiencing various problems. Say, for example, our population of the Earth is continuously increasing, and these increasing population need a lot of resources. Say, for example, food, and we do not have the sufficient food to feed this global population. And in order to increase the more and more food, we are adding a lot of fertilizer and pesticides into the soil, which ultimately are turning these type of the fertile land into the barren land. These increasing the population will also need a different resources. Say, for example, school, houses, clothes, all these things. In order to produce all these things, we need to utilize more and more fossil fuel, more and more urbanization, and more industrialization. All these things will actually lead to release of the higher carbon dioxide and other greenhouse gases into the environment, which ultimately cause the climate change. So throughout the globe, various researchers are working on to identify various solutions, sustainable solutions to solve this problem. In this regard, one of the better solution to produce the fuel is utilization of the various type of the biomass to generate the fuels. These biomass actually are categorized into the 3 different categories. One is the first generation biofuel production crops, which is majorly produced from oil-rich crops and sugar crops. As I have mentioned in my previous slide, that we do not have a sufficient food to feed the population of the overall globe. In this scenario, utilization of these food crops to produce the fuel is not a good option. So we cannot consider these as one of the very good resource to make the biofuel. Next, very good option is to utilize the second generation biomass, which is majorly made up of the lignocellulosic biomass. So this is once we harvest our crops, the remaining residue in the crop plant, we can say it's the lignocellulosic biomass. Various research groups throughout the globe is basically working on to utilize these biomass to make the fuel. But one of the major problem that they all face is the composition of these biomass. So they generally contain cellulose, hemicellulose, and lignin. Apart from cellulose, hemicellulose, and particular lignin are very complex molecule. So in order to hydrolyze lignin, mainly people are utilizing the acid or alkali or high temperature pretreatment, which type of treatment is generally producing certain inhibitory compounds, or sometimes they are also produce the 5-carbon sugar, which is basically not readily utilizable by the most of the fermentative microorganism. And the inhibitor which is actually produced from this lignocellulosic biomass are basically like inhibitor for the organism. So it's not a good option, but though throughout the globe, various researchers are working on it because it is, produced in a very high quantity, and utilization of enzyme is actually now considered as one of the very good option for the pretreatment to convert the biomass. I basically work on the third generation biomass, which is majorly made up of the macroalgae and microalgae. I am mainly focusing on the microalgae as a source of the biofuel and bioproducts. Why I am considering microalgae as a source of biofuel or industrial products? Because microalgae generally have a very high growth rate, and we can say the cost of the production is low. Why I'm saying cost of the production is low? Because you can cultivate microalgae in away from farmland. So if I am cultivating the algae which is in the land which is not utilized for the agricultural purpose, I am not destructing any food chain, and then I'm also not into the food versus fuel debate. And many of the algae have ability to utilize various type of the seawater, brackish water, wastewater, or sewage. Okay, and they also have ability. Basically, they are the photosynthetic system, so they are fixing the atmospheric carbon dioxide. But many of the algae have ability to obtain the CO2 from the power plants, which also contain the SOx, NOx, and all these things. So if I am utilizing a microalgae, and if I am using a sewage, wastewater, or the flue gases to cultivate them, I am not only reducing the environmental footprint, but I am producing the biomass which has some economic value. So we can say that this type of thing is actually a win-win strategy, where we are cleaning the environment. At the same time, we are also producing the valuable biomass which can be used for a different application. And many research indicate that the oil productivity of the microalgae is generally very high compared to the oil rich crops such as sunflower. And another very interesting thing about the microalgae, as I mentioned, in case of the lignocellulosic biomass or a crop plant, they have the lignin component in them, while microalgal cell do not contain lignin. Mostly they have the cellulosic cell wall, and their cytoplasm is full of starch. So if we are utilizing a de-walled biomass, it means we have already broken down the cell wall. Now the cytoplasm, which is full of the starch, we can easily hydrolyze using a simple normal mild acid, or maybe we can add amylase to convert them into the fermentable sugar to produce the bioethanol. Microalgae also have a very high amount of the protein, so it can be also used as animal feed, or once we extract the lipid, these biomass can also be used as a organic fertilizer, because they have a very good nitrogen phosphorus ratio, which can be used as a organic manure or as a fertilizer. So considering all these advantages, I consider to work on the microalgae to produce a fuel. Now, as I'm talking about TOR, so first of all, I will explain what is TOR and why I'm interested to work on TOR signaling to improve the starch content. So TOR is a target of rapamycin. So this is basically a drug, rapamycin, which inhibits the kinase. So the drug is actually isolated from the Easter island known as Rapa Nui. From this name of this island, the drug name was given rapamycin. It is actually isolated from actinomyces, and this is the structure of the drug rapamycin. So this is the structure how the TOR is look like. So they have these N-terminal heat repeat, FAT domain, FRB domain, a kinase domain, and FATC domain. So if we consider the yeast and the mammals, generally they find two different TOR complex, that is TOR complex 1 and a TOR complex 2. TOR complex 1 is specifically inhibited by the drug rapamycin, while the TOR complex 2 is insensitive to rapamycin. And they have these different protein complexes associated with the TOR, that is Raptor, LST8, Richter, LST8. And it has been revealed in case of the mammals and yeast that TOR is involved in various cellular processes, starting from protein degradation, autophagy, ribosomal biogenesis, transcription, translation, cell cycle regulation, cytoskeletal organization. Particularly TOR complex 2 is mainly involved in the cytoskeleton regulation. However, if we consider the higher plants and the green algae, so far only TOR complex 1 is identified, and there is no information about the TOR complex 2. So in order to understand the role of TOR signaling in the starch accumulation, I have used an organism which is known as Cynidiocyzone marole. So basically, this is a red algae. It's a very interesting model system. So far, no one is, working in India, but I have recently started to work on this. Why I'm saying it is a very interesting organism. So this is an eukaryotic thermoacidophilic red algae, and the interesting thing is that the complete genome sequence of this organism is readily available, and various molecular genetic techniques such as transformation, tagging, construction of knockouts is very well available. Even we can do the homologous recombination, and the genome size of this organism is very small, around like 16.5 MB with like around 100 transcription factor. And one of the interesting thing about this system is that generally in the cell we have one nucleus and multiple chloroplast and multiple mitochondria. But in these organisms, in a single cell, we have a single chloroplast, single mitochondria. So the people who is basically interested in organelle division or maybe the retrograde signaling, that is the signal from either the chloroplast to nucleus or a mitochondria to nucleus, maybe this system is one of the very interesting system. So though this system is very important, as I mentioned, in case of the plants, mostly the plant people face some of the problem when they are working on TOR signaling. One of the problem is that the FKBP12, I mean, the structure of the FKBP12 protein of most of the plants and algae have a different structure than the yeast and mammals. So these FKBP12 protein is actually unable to form a complex with the rapamycin, and when this protein form a complex with rapamycin, it actually inhibiting these kinase domain. So because the structure is different in the plants and algae, the people who are working on the TOR signaling in plant and algae generally need to express the yeast FKBP protein or the mammalian FKBP protein into their system. So before actually I joined the lab in Tokyo Tech, my postdoc mentor has actually expressed the yeast FKBP12 protein into the C-zone cell and make this cell rapamycin sensitive and give the name this strain as SF12 strain. So in my next slide, I will explain the use of these SF12 strain to understand the role of TOR signaling in starch accumulation. So why actually I thought to work on a TOR signaling and starch accumulation. So actually in 2015, my postdoc mentor, Professor Sausuke Yamamura, has published one paper when he mentioned that when you add a rapamycin to this SF12 strain, the strain is accumulate very high amount of the triacylglycerol. It is like almost comparable to the nitrogen starvation condition. And these triacylglycerol can be easily converted into the biodiesel. So when I saw this paper, I thought that because in the cell, starch and tag is both the competing pathway, and actually because of the less reduced state of the starch than the triacylglycerol, probably this might accumulate very early compared to tag if I inhibit the TOR through rapamycin. So we propose that can we check what happened when we inhibit the TOR into the C-zone, and can, if we improve the starch content, maybe we can convert them into the bioethanol. So we proposed this proposal to JSPS and we got funding from them. Actually, it is also reported in other organisms. Say, for example, in case of the Arabidopsis, when actually you inhibit the TOR, cell is accumulating very high amount of the glucose. And in case of the Chlamydomonas, it also resembles that inhibition of TOR will accumulate very high amount of the starch into the cell. It is also there in case of the mammals and yeast. I have not mentioned in the slide because I'm mainly focusing on the photosynthetic system. So after getting the funding, I thought, let me first try the experiment to inhibit the TOR using the rapamycin in the C-zone cell and estimate the starch. So as you can see from this slide, that increasing the incubation with the rapamycin, we show the significant amount of the starch accumulation after the 12 hour of the rapamycin addition, and it is like almost 10-fold higher compared to the drug vehicle DMSO. So we can say that the similar to the yeast, mammals, Arabidopsis, and Chlamydomonas, C-zone is also accumulating very high amount of the starch under the TOR inactive condition. Many researchers, those who are working on the biofuel, they are generally doing the nitrogen starvation to improve the starch or a lipid content in the microalgae. So I have done the another experiment where I have added the rapamycin. I have also provided the nitrogen starvation to the cell. And then in the third condition, I have done the nitrogen starvation along with the rapamycin treatment and estimated the starch content. And as you can see from this slide, compared to DMSO, all these three conditions are producing a very high amount of the starch. In fact, the amount of the starch in the rapamycin and the rapamycin with nitrogen starvation is not significantly different. It means that TOR might be a major regulatory pathway for the starch accumulation in the C-zone. So this is true that TOR is like involved in the starch accumulation. But as I mentioned, there is no molecular mechanism available even in case of the yeast and the mammals. So we thought that let's try to uncover what might be the mechanism behind these processes. So at that time, actually, the microRNA data was available under the rapamycin treatment. So as a first step, I have checked whether any transcript related to carbohydrate metabolism is affected by the rapamycin treatment. And to my surprise, I do not find any transcript which change its, significantly change after the rapamycin treatment. It indicates that there is a possibility that the starch accumulation by TOR might be a post-transcription level. And it is also like evident because TOR is basically a kinase. Maybe it regulates its substrate through the phosphorylation, dephosphorylation mechanism. So in order to uncover this post-translational regulation of the starch, we have collaborated with the Tohoku University and performed the phosphoproteome under the rapamycin treatment. So this is the basic steps that they have performed in order to get the phosphoproteome. We have done the phosphoproteome at 4 different time points. That is the 2, 4, 8, and 24 hour of the rapamycin treatment. And we actually identified total 52 protein which change its phosphorylation. These proteins, if we categorize them, they are involved in the amino acid metabolism, cell cycle regulation, protein binding, RNA binding. Many of them are involved in translation, carbohydrate metabolism, RNA degradation, and many others. And we also have identified that most of the residue which is phosphorylated during the rapamycin treatment are basically the serine. Only one protein which regulates at a threonine level. Then we perform the motif analysis that what are the motif which is regulated. So we identified that RXXSP, that are the motif that we have identified. When we compare the motif with already available motif for the eastern mammals, these are the exact same motif. So we can say that maybe the motif which is regulating through which TOR is regulating might be conserved even in the photosynthetic system. I would like to say that we have published this paper in Plant Journal in 2019, and this is basically the very first paper in the plant lineage who actually unrevealed the role of TOR signaling through a phosphoproteome analysis. After that, in 2020, one paper was published in Nature Plant after we published this work. So, as I mentioned that we have identified almost 52 protein involved in the different processes of the cell. We just focus only on the carbohydrate metabolism, and out of 52 protein, almost 13 protein are actually involved in the carbohydrate metabolism.
Sir,
And
Mr.
here,
Mute your mic. Sir, Mr. Sahu, mute your mic. It should not again unmute. Please
so
continue.
yeah, so actually we basically now focusing on the two protein. One is known as CMK020C, which is actually in the database denoted as a similar to glycogenin, and we have given them a name CMGLG1. And the another protein is CMM317, which is denoted as a probable starch or a glycogen synthetase, and we denoted it as a CMGS. So this is the schematic how basically these two enzymes is actually work. So actually this is very similar how our PCR system will work. So these CMGLG1 is basically a primer. You consider it as a primer in the PCR reaction. It actually initiate the glycogen synthesis and add almost 20 residue of the glucose to the growing chain. And then the CMGS, maybe you consider it as a DNA polymerase, which actually continuously adding the glucose residue into the growing chain and actually make the starch residue. Because these two enzymes are very critical, we thought that first try to work on these two protein. So at the very first step, we thought that let's try to overexpress them in the cell. And if they may involve in the starch accumulation, maybe we will get a higher accumulation of starch in the transgenic lines. So we have constructed the CMGLG1 overexpress as well as CMGS overexpress cell lines, and then we have also checked the starch content. And as you can see, compared to the control vector and the CMGS overexpression line, the CMGLG1 overexpression line is accumulating significantly higher amount of the starch. So we thought that let's try to focus now on the CMGLG1 and check how it is actually regulating the starch accumulation through TOR signaling. So because as I mentioned that CMGLG1 is a glycogenine type of the enzyme, and if you consider yeast generally contain two glycogenine enzyme, glycogenine 1 and glycogenine 2. And if you knock out both the glycogenine from this yeast is actually unable to accumulate the glycogen, and they have given the name CC9, the double knockout strain. So what I did, I actually expressed the CMGLG1 into these glycogenine double knockout strain. And as you can see from this panel, this is the wild type yeast strain accumulating glycogen. This is the CC9, which is basically the double knockout by which is expressing the CMGLG1. You can see that we are actually partially rescuing the glycogen accumulation compared to these not empty vector, because it is like yellow. It means it is not producing. This is the very simple technique. We are just exposing the yeast colony into the iodine vapor, and depending upon the amount of the glycogen, the colony turn into the brown. We have also checked the residue, tyrosine residue, which is actually involved in the self glycosylation into the uh, glycogen molecule, and we identified that the CMGLG1 actually have these conserved residue. This is the secondary structure of the glycogenine CMGLG1. It contains actually transit peptide, 20 amino acid transit peptide at the N-terminal site. They have the glycosyl transferase domain, which is actually transferring the sugar molecule, and actually it has a 5 transmembrane domain at the C terminal. And this is the serine 613 residue, which is actually phosphorylated by TOR signaling. And we also have to confirm that the when we express basically these glycogen, we actually find a 2-band into a western blot. We thought that maybe the upper band might be due to the phosphorylated glycogen, and lower is maybe the non-phosphorylated band. So in order to confirm that, we have performed the lambda phosphatase inhibitor treatment, and we do not find any change in the 2-band in all the conditions, which indicate that maybe this upper band is maybe due to the premature glycogen, because actually it contains these transit peptides. So maybe peptide might cut. That's why maybe we have a double band here. It is not the phosphorylated glycogen. So in order to confirm that the serine 613 residue is basically the key residue which actually important for the accumulation of starch, we need to confirm this by constructing the phosphomimic and the phosphodimimic strain, where we are changing this serine 613 to aspartic acid to construct it is to the similar to the phosphomimic strain, which is like a continuous active state, and then phosphomutant in which will convert the serine into the alanine residue. So we have constructed these strain CMGLG overexpression, CMGLG overexpression, where we have converted the serine residue to aspartic acid, and here serine residue to the alanine. And then we have measured the starch content in all the three conditions. And as you can see, compared to the wild type overexpression and the phosphomutant overexpression, here we observed almost, compared like 60% reduction. So which indicate that the serine 613 residue is very critical for the activity of the glycogenine. And we have observed that this residue changes phosphorylation through the TOR signaling, which actually confirm that TOR is actually involved in the starch accumulation in the C-zone cell. So if we propose a model which is something look like this, that when TOR is active under the normal growth condition, CMGLG1, particularly the serine 613 residue, is phosphorylated and cell will not accumulate a very high amount of the starch. However, when we inhibit the TOR through rapamycin or maybe through the nitrogen starvation condition, this TOR become inactive. The phosphorylation status of this glycogenine is changed. It become a dephosphorylated one, and then cell will accumulate very high amount of the starch. We can utilize the starch to produce the bioethanol. In fact, we have collaborated with other our colleagues in the chemistry department, and they have developed some SN catalysts to make some sort of sodium valerate or that type of molecule which have some pharmaceutical relevance. I'm not sharing this story here because here my interest is to work or explain my work in the glycogen. So as I have mentioned in my previous slide, that in the yeast, basically they have the two glycogen 1 and glycogen 2. If you remove glycogen 1 from the yeast cells, yeast cells still have ability to accumulate the starch. But if we knock out both the glycogen from the yeast, yeast is unable to accumulate the starch or the glycogen into the cell. So I was curious that what might be in case of the C-zone, whether it also contained another glycogen. So actually I mined the genome, and instead of 2, I actually identify 4 different proteins similar to glycogen in the C. marole. One protein in which I have already explained, that is the similar to glycogen, CMGLG1. Another protein is CMR358C, which is actually involved in the plastid division ring, which is denoted as a PDR1. And the third one is CMJ262C, which is actually the mitochondria division ring. So at that time, there is one professor at the University of Tokyo whose name is Yamato Yoshida. He actually published this paper, I think this PDR1 in a Science and MDR1 in a PNS, where he mentioned that during the organelle division, like division of chloroplast and mitochondria actually form one polyglucan filament, and after that the organelle is dividing. So they have performed actually the proteome analysis to protein which is formed in that particular ring, and they have identified this glycogen type protein which is involved in the organelle division. But when I contacted Professor Yoshida, he told that, Imran, I didn't identify either the glycogen into my the protein which I have actually extracted from the nanoglucan or a polyglucan ring when it is like forming for organ division. And so it indicates that maybe this protein has a different role than the organelle division. And I actually identified fourth protein, which is actually CMG174C, which is also denoted as similar to glycogen glycosyl transferase. So I thought that these two proteins are involved in the organelle division. The protein which I have worked for the TOR signaling is a glycogen, and this is a glycogen glycosyl transferase. Maybe this is maybe the glycogen 2 for the C-zone cell. In order to understand more about these protein, I have actually overexpressed these protein. First I will explain about the phylogenetic. So I have constructed basically the phylogenetic analysis, and I come to know that CMGLG1 is actually clustered with the Arabidopsis and plant, glucosyl glycogen transferase, while actually this CMGLG2, which is actually this glycogen glycosyl transferase, is actually clustered with yeast and rabbit muscle glycogen, which is like very well known, very well studied glycogen. So there may be possibility that this CMG174C is actually the main glycogen for the C-zone cell. Then I have actually, similar to glycogen 1, I have overexpressed this CMGLG2, and you can see the transcript level, which indicate that the strain is properly constructed. We have also confirmed the CMGLG2 using the Western blot. Basically, we have expressed the flag tag at the C terminal, and using the anti-flag antibody, we have confirmed the proper construction of the CMGLG2. And finally, we have also estimated the starch content, and it is also accumulating starch, but the amount of the starch is not that high, similar to our GLG1. So there may be possibility that there is some different mechanism. The growth of this strain is also comparatively slow compared to this CMGLG1 overexpression strain, and it is also the amount of the starch which is accumulated by this strain is also a little less. So I mean, there may be possibility that some other mechanism is there, because so far in our phosphoproteome we have not identified a CMGLG2. So if I sum up all these things, we can say that in case of the C-zone cell, they have the four different glycogenin type protein. One is CMGLG1, which is the glycogenin type protein, and it is regulated through the target of rapamycin signaling. There are two more protein. It is the PDR1, which is involved in the plastid division, and then MDR1, which is involved in the mitochondrial division, which is not regulated through the TOR signaling. The fourth protein, CMGLG2, we have not identified it in our phosphoproteome. Neither Professor Yoshida has identified during the organelle division purification. So still we don't know what might be the regulatory of the CMGLG2, and we have recently initiated some of the work with the CMGLG2, that what might be the regulator for these and how it is contributing into the glycogen accumulation into the cell, and can we overexpress both these protein into the cell to make the powerful strain which accumulate very high amount of the starch, so that we can convert these starch into the bioethanol, or maybe we can produce some industrially relevant product out of it. So I think that's all from my side. And the story that I have mentioned is actually the postdoctoral work. I mean, part of the postdoctoral work which I actually done with my postdoc mentor Sausuke Imamura. Earlier, he was at a Tokyo Institute of Technology. Now he actually moved to NTT Docomo, and my lab is actually funded by funding from DST SERB India, as well as the GSBTM and the Gujarat Biotechnology University. And I'm thank you very much to all of you, and happy